All right, welcome everybody to this month's StaPH-B call. Today we have Lucy Lee and David Dynerman from the Chan-Zuckerberg BioHub. They did a close collaborative project with the Alameda County lab in California, where they started sequencing CRE isolates in that laboratory, but lacked the sort of bioinformatics capabilities to do proper analysis. So the collaboration with the Chan-Zuckerberg BioHub was to develop this project or this platform called Madrone to sort of meet those bioinformatics needs. And from that short intro, David, Lucy, I'm going to go ahead and give you the floor and mute my mic from here on
out. Thank you all for attending. We're really excited to get the opportunity to present to you. So my name is David Dynerman. I'm on the data science team at the BioHub, and my colleague is here.
I'm Lucy Lee, also on the data science team at the BioHub.
So Lucy and I have been leading a project called Madrone, which is a project to try and bring sort of some advanced genomic epi tools into routine use at the small scale level. So we're excited to tell you about it. Let me also mention, just in case people aren't familiar with the BioHub, so the Chan-Zuckerberg BioHub is a nonprofit biomedical research institute in San Francisco, affiliated with UCSF, UC Berkeley, and Stanford. So we're a philanthropic organization, but we're engaged in basic and translational research, and we are a nonprofit. Okay, so let me kind of introduce sort of the motivation behind Madrone, behind our project. So local health departments, especially in the state of California, these are the entities that are tasked with tracking and preventing the spread of infectious disease, really, on the ground. And if we throw in hospital epidemiology and hospital infection control units, these are the sort of frontline organizations that are tasked with preventing hospital-acquired infections, tracking the local spread of antibiotic resistance, especially in the Bay Area, tracking the introduction of new antibiotic resistance genes from other regions of the world. So, despite the fact that these local units have, this very important task, they often lack capacity in bioinformatics and statistics, math and modeling, and computational infrastructure. So this combines to often create the situation that infection control and public health decisions at the local level have to be made on the basis of pretty primitive data analysis. So these are public health nurses basically looking at spreadsheets of diagnostic lab results and trying to make the best guess possible just on the basis of eyeballs on a spreadsheet. So the goal we set out with Madrone, with our project, is to try and improve the tools that are available on a routine basis at the local level. So we want to bring some of these advanced genomic epi tools that the community has developed, including many people on this call, into routine use at the local level and at the hospital infection control level. So we're targeting public health nurses, infection control nurses in the hospital, at the county health department, versus developing tools that might be used at CDC or at the WHO or something like that. So I should also mention to please interrupt for questions at any time, and also we'll hopefully have a lot of time for discussion afterwards. All right, so to put kind of a cartoon to all of this. So at the local level, data is generated. So this might be clinical microbiology lab culture and results. It might be patient movement in a healthcare facility. And analysis has to take place on the basis of, here, let me display this slide. So analysis has to take place on the basis of this data. And again, the analysis that we see even at academic medical centers like UCSF, frequently is limited to human beings looking at spreadsheets, and similarly at the Alameda County Health Department. On the basis of this analysis, the public health nurses or the infection control nurses, the public health epidemiologists, they have to decide to take certain actions. So they have to decide, are we going to put patients on contact precautions? Are we going to order environmental decontamination? Are we going to remind are we going to have a training session where we remind the physicians to wash their hands a lot? So these concrete actions have to be taken on the basis of this, primitive data analysis, basically manually examining spreadsheets. So our conclusion is that limited analysis, the basic constraints of these basic spreadsheet analyses cause limited action. So you're not able to take, the fine grain action that might help infection control. You're not able to take the right action at the right time. So we feel that that this kind of observation that we made was also, very nicely summarized much more rigorously by the CDC in an MMWR from 2018. So this is a survey of all public health, a survey that was sent out to all public health entities in the United States, including, these like overseas US territories like the American Virgin Islands and, American possessions in Samoa and stuff like that. So really, they sent it everywhere. And from this survey, the CDC concluded that in public health, in American public health, there is an ongoing unmet need for additional epidemiologist positions in infectious disease and also informatics, and highlighted that serious capacity deficits remain, especially in informatics as well as genomics. Importantly, also, the CDC concluded that despite a hiring boom in public health over the last 10 years, the capacity deficits in informatics especially remain. So the CDC is making the point in this MMWR that just hiring more staff at the local level, giving more federal funding for more positions at the local level is not necessarily going to translate into capacity improvements in informatics, especially. So we really believe that this indicates that the time is ripe for perhaps some kind of alternative approaches, and so that's what we're trying to develop with Madrone. Is we're trying to develop a end-to-end platform for some more advanced analytics that helps inform the actions that local public health officials and local infection control providers might be able to take. Okay, so that sketches sort of the motivation for us. So next I'll describe the project. Any questions so far? Please interrupt at any point if there's questions. All right, so the way the system works right now, and so the system was developed together in close collaboration with the Alameda County Public Health Department, and this system is in routine use now at the county health department. So it took maybe about a year to move from sort of the one-off prototype phase to the time, to the place we are now where they're actually routinely using it on an ongoing basis every week. All right, so Alameda County, due to a CRE outbreak in 2015, implemented a local health officer order that requires surveillance and the submission of CRE cultured isolates to the public health lab. So anywhere in Alameda County, if you're a diagnostic lab, if you are a healthcare facility, if you're a hospital, if you're a nursing home, if you culture positive, a CRE bacteria positive, you must send that sample to Alameda County Public Health Lab. So the public health lab then sequences these isolates, and that's where we come in. So after sequencing, the staff from the public health lab drag and drop the sequences onto the Madrone web interface. We then initiate a backend bioinformatics analysis where we analyze the pathogen genetic sequences, and then we generate reports for them. And these reports, we'll go into a little bit more detail on what they look like, but these reports were again generated with the infection control public health nurse user in mind. So they were generated to let them act more effectively in their workflows. So these reports are delivered and examined and interacted with by public health nurses at the Acute and Communicable Disease Division of Alameda County Public Health Department, and then on the basis of that, they decide whether or not to take action. All right. So let me tell you a little bit about the bioinformatics backend, because I know that a lot of StaPH-B has a lot of world experts on this kind of genomic epi analysis. So we have a pretty simple bioinformatics backend. So the input is going to be whole genome sequencing reads of bacterial cultured isolates. We do antibiotic resistance and MLST typing using SRST2, which is a tool from Kat Holt's lab. Then for the SNP distance calculations, we use a pipeline that we actually wrote, which I'll get into in more detail on the next slide. It's called SPID. So this is an alignment-based SNP calling pipeline. We use Minimap2 for the alignment, some SAM tools, pre-processing, and then the SNP calling is done in a set of Julia scripts that we wrote. Once we have those distances established, we use RaxML to generate trees, and the overall output of this pipeline are the SNP matrices, the trees, which AMR is present, which antibiotic resistances are present, and the MLST sequence type. And all of that, so this is the output of the backend, but then of course the important thing is that this backend output is visualized in a kind of easy to use fashion for the public health staff. All right, so why did we write our own SNP caller SPID? So basically this came about based on feedback from the health department. So with traditional joint callers, like GATK and others, as you add more samples, the sort of relevant core genome size shrinks. And moreover, joint calling may slightly change previous results. So in other words, you might have sample A and B, when you first compute it, they get a SNP distance of 10. But then you add a couple more samples and the core genome shrinks, and now A and B are maybe 13, like that SNP value changes. And that was very disturbing for the health department users. They were like, well, we thought sample A and B were 10, and now all of a sudden they're 13 or they're 7, right? And so those slight changes don't fundamentally alter, the sort of like large scale conclusions, but we wanted to engineer a pipeline that they were comfortable with, and that didn't have this sort of instability. So SpitJL is an online reference-based alignment pipeline. So what I mean by online here is that as you add additional samples, the previous distances between previously computed samples don't change. So the system is online, so it's basically built for continuous updates. So the idea is we can cheaply add additional samples to the tree without recomputing all the pairwise distances jointly. It's stable so that distances don't change with new samples. And we have done a round of validation, against the traditional SNP callers, and the results agree well. So SPID.jl, this backend, is available. It's open source under the MIT license, so it's available on this GitHub link at the bottom of the slide, and we can send that around also via email later on.
So sorry to interrupt. I got a quick question for you. So when you're calling your SNPs, are you having a defined set of reference sequences you're using that don't change based on the organism, or are you continually revalidating or re-updating reference sequences uses for your SNP calling?
So right now, samples are collected into reports based on the species and the submitting organization. So there's like a Klebsiella pneumonia tree for Alameda County. And we've chosen a single reference for each of those reports. But the Acinetobacter baumannii tree for Alameda has a different reference than the Acinetobacter baumannii tree for UCSF. So okay. Those are sort of separate reports, and we've been choosing the reference that most closely matches the samples that are being submitted.
And I think like moving forward, we want to move to a system where we have like more fine-grained reports than just at the species level. So for example, for Klebsiella pneumoniae, there's so much diversity, sometimes even within the same sequence type. So I think moving forward in the future, we want to have an automated pipeline to select and to cluster the samples and group them into reports at levels below the species level.
Cool, thank you.
And let me maybe also just go through this cartoon flowchart on the right of this slide. I apologize, I should have gone through it on the previous slide. So the raw reads are coming in on the top, and then on the left, they branch off just into SRST2. So this is a tool that just uses bowtie to basically call MLST types and flag known AMR genes. So that's the left branch. So the right branch, we use Minimap to align the reads against the references. We use SAM tools to generate a pileup, and then we use these Julia scripts that we wrote to call Snips based on the pileups. And we have some kind of basic filtering criteria, like we require, I believe, at least 10 reads piling up at a site with 90% concordance to call a SNP. And then we use, as I mentioned, RexML to generate the trees for the reports. Okay, all right, so that's the back end. So what about the front end? So, our goal, kind of like I mentioned before, is, we envisioned kind of from the beginning for this to be a very translational project, right? So we didn't want to just somehow, develop a set of backend scripts that then they needed to, have command line expertise to run. We really wanted to deliver something where the public health nurses at the health department can log in and, and they can see the data without having an expert interpret it for them. So we were really setting out to engineer ourselves out of the loop, right? So there's this loop where Alameda submits data and then their public health nurses interpret the data. And we wanted to remove ourselves as much as possible from that loop. So the goal was to make the front end, usable by these end users. So in particular, we focused on quickly summarizing aspects of this backend analysis that they can actually act on. So in particular, sort of like Lucy alluded to, big giant sort of super trees showing like all Klebs pneumo samples for the last 10 years in Alameda County are really cool to us because we're like, oh, look at all the patterns and how they change over time. But the public health nurses can't really act on a tree like that, right? What the public health nurses are interested in is what's happening right now over the past few months. How are the trends changing? Is there an outbreak happening that we need to intervene in or investigate further, and so on. So we really focus the front end on those kinds of that kind of information because that's what they can act on. Another important thing is we wanted the reports to be interactive so that those end users who are maybe a little bit more into the data side, the quantitative side, they could really dive deep and spend more time interacting with the reports, doing hypothesis testing. Is this a cluster? Is it not a cluster? And so that interactivity was an important sort of design goal for the front end. So what does this look like in practice at Alameda County Public Health Department? So the main way that they found the tool useful is to help them in their cluster calling process. So that means that on the trees, they wanted to easily visualize closely related samples. So closely genetically related samples, because those would be suggestive of a possible outbreak cluster to follow up with. But we also, in that cluster visualization feature, we wanted to allow users to experiment with different cluster thresholds to kind of hypothesis test, as they're cross-referencing it with the private data that they have on their end, as well as to triage potential clusters based on the antibiotic resistance patterns. So those were kind of the goals we identified in terms of what would be useful for the public health nurses. Okay, so here's a video, if it starts playing, of, oops. Yeah, there we go. So here's a video of the interface. So up above, sorry, I'm new to videos. So up here on the top, you see that when the user logs in, suspected outbreak clusters are flagged for them and named based on the facility. So they're able to sort of immediately see like, okay, at County Hospital A or B, there's a potential outbreak that I need to dive deeper on. So they can interact, they can click on those suspected clusters to be taken to them. On the tree itself, the suspected cluster that was selected up above is highlighted in red. Other related samples that satisfy the threshold are highlighted in gray. Additional sample information as well as which AMR gene is visualized here at the bottom for those samples that are selected, and a SNP matrix is dynamically generated so that they can interact with the tree, select different samples, and then view the SNP thresholds. So what they use that for frequently is, if they have sort of suspicious patients, they'll select them on the tree and then see how the SNP distances play out. If they, by what I mean by that is, if they have reason to believe that a group of patients might be transmitting to each other, they can select those patients and then view a restricted SNP matrix to those individuals. So let me also just mention that on the right here, we visualize the antibiotic resistance as a heat map. So this is these black and gray squares on the right. So the columns of this heat map are known antibiotic resistance genes. There's a black square if that gene was detected in that sample. And as you hover over them, you can get more detailed information about the specific alleles that were picked out for each of the antibiotic resistance genes. All right. So let me...
if I can ask a quick question
Yes.
here. So you did a great job to sort of like have the visualization about specific antibiotic genes. I'm just curious about like how the nurses and health practitioners feels like in terms of the presence and absence or potential mutations with those specific genes and how that related to specific antibodies. I'm curious about your thoughts in terms of translate those kind of gene-specific information into like specific drugs.
Yeah, so Madrone is not a clinical tool, right? So these types of genotypic predictions or genotypic flaggings of AMR are not meant to be used for like clinical intervention. Like if you saw that something was whatever had a beta lactamase here, you wouldn't want to switch to a different carbapenem class antibiotic. This is not a clinically validated tool. Like any information here, is to be used for kind of public health and infection control purposes. So what the nurses are doing with the antibiotic resistance readout on the right is they're looking for global patterns and public health patterns. So basically, they have they are constantly looking at the antibiotic resistance that's been seen in healthcare facilities in their jurisdictions. And they are, so they know what's endemic and what's not. So they're looking for the introduction of new high-risk antibiotic resistance genes that haven't been seen previously. And they're also looking for, changes in the patterns that are being seen. So for instance, if there's a small number of, OXA1 samples over a few months, and then all of a sudden they get 5 in a row, they would want to dig in deeper into those 5 cases and see, is there something going on? Are they all at the same healthcare facility? So it's that kind of intervention. It's not patient treatment intervention.
Good to know. Thank you.
Okay, so the status of the project currently is we have, we've analyzed 850 samples from a bunch of different places. So Alameda County is the user that's using it actively on a weekly basis. But we've spoken to a lot of other facilities and we've analyzed samples, for a bunch of other hospitals and public health departments. And, we're hoping to bring additional public health departments on board with the tool. So as I mentioned, it's in routine use at the Alameda County Public Health Department, and very excitingly, they've seen an improvement in investigation efficiency by 33%. So what they did is they looked retrospectively over the past quarter, and they compared the investigations that they undertook based on Madrone versus the investigations that they would have undertaken under their pre-Madrone workflow. And what they found is they found that Madrone, mostly through ruling out clusters that they would have otherwise investigated in the absence of whole genome sequencing data, genomic epi, they would have investigated an additional 33% of cases. So this is a huge improvement for them, because Alameda County is a county of almost 2 million people, and they have an ACD staff of 4. So for the, as you can imagine, those 4 people are incredibly overworked. Their informatics systems are not tied to the healthcare facilities. So when they investigate a case, they have to pick up the phone, they have to get faxes of people's medical records, they have to pore over the faxes, looking for patterns. So to save them 33% of cases that they immediately know are not outbreaks because they're tens or hundreds of thousands of SNPs apart is a huge, huge benefit from them. And that's a benefit that we would like to replicate at other places that, that might gain from it. We also, we're really happy we've had strong user engagement from it. So among that ACD staff of 4, we see the full spectrum. 1 or 2 of them are really into nerding about the data and drilling down, and we get emails from them late at night saying, like, how come, like the triangle inequality seems to be violated for these 3 samples? What's going on? And then we have, 1 or 2 that are finding the tool very useful, but appreciating the fact that the interface is simple and easy to use.
Actually, just adding on to that, I think definitely seeing this in routine use and like weekly use at Alameda County has been like our greatest pride with this project. And actually, we just got an email from the Alameda County team where they said they now have a weekly drone keeper rotation in which they check on the latest whole genome sequencing and Madrone results to help them document potential clusters and prioritize their scope and intensity of their investigations. So definitely having them on board to help us develop this tool and seeing them like part of their job description, their job workflow now has been great to see.
Yeah. Okay, so our main goal for 2020 is to try and expand and build on this success. So specifically, again, sort of the way we envision this project is about making kind of complicated data analysis usable at the peripheries of the public health system. There's a lot of expertise in public health bioinformatics in the United States, as you all know, right? If there's a serious large-scale outbreak where hundreds of people are sick, the state public health department has experts, the CDC has experts, this data is going to get analyzed, the genomic epi is going to get done. What we're interested in is those outbreaks of 3 patients who have an HAI. We're interested in those small-scale local interventions at the periphery of the infection control and public health system, where we feel that these tools can really make an impact if they're automated and if they're easy, if the results of these analyses are easy to use. So in mind, in that spirit, we're really interested in getting some new local health departments or state health departments if they feel that their public health nurses could use a tool like this. We're really keen to get some additional health departments using it routinely the way Alameda County is using it routinely. A separate module that Lucy is leading up is the implementation of an electronic health record-based component to this for hospital infection control. So we're working with UCSF to basically get their infection control team using Madrone routinely, but where they're visualizing not only genomic data, but also spatial and temporal data from their electronic health record. All right, so I just wanted to end by just highlighting the great team that we've had working on this. So in addition to Lucy and I, we have Jack, Phoenix, Aaron, Sam, and Chaz, and they've all put a lot of work into this, and we're very fortunate to kind of put together such a great team here at the BioHub. Okay, so thank you very much for your attention, and we're happy to sort of delve into a lot more detail now in the remaining time.
Awesome. Thank you, David. Thank you, Lucy. Definitely interested in opening it up to questions after that. I kind of wanted to start it off, if that's all right. So those reports look super helpful, and I think you had already mentioned that the backend is something that we all have access to through that public repository. I think at one point you had mentioned in a different conversation that there was works to make that front end interface also publicly available. Can you speak on that? And especially because I think a lot of public health labs are going to find this incredibly useful, but data transfer is going to be a major concern, especially to get utilities like, it looked like there was some sort of Google Maps feature there too, to sort of get the metadata to the samples included. So that'll definitely be a big challenge for states to sign off on. But getting that sort of same system set up locally may be a more feasible approach.
Yeah, absolutely. So open sourcing, so we have a blanket policy to open source everything that we produce. So open sourcing the front end is a high priority. Currently, it's still kind of in a state where it requires sort of a fine touch by us, the people that wrote it. So basically, we just need to put some effort into documenting how to get it set up. But long story short, it's a high priority for the first half of 2020. To get it all open source. We definitely are, we're sensitive to the kind of data sharing issues you mentioned. So the kind of like on-prem version of this is definitely something that is doable, and that we're interested in documenting how to do that. I will, however, mention that one thing that is nice about pooling data together is the possibility to observe cross-jurisdictional information. So Madrone has built into it kind of a differential level of access to the front-end reports. So what that means is that it's possible for San Francisco DPH to submit samples, and Alameda County to submit samples, and for those samples to be on the same tree. But when a San Francisco user views them, they don't see any of the sensitive Alameda data. And when Alameda views that same tree, they don't view any of the sensitive San Francisco data, which would just be like maybe the facility, like the hospital that the patient's at. And the benefit there is that, patients move between San Francisco and Alameda, of course, Alameda County all the time. So it's certainly possible that they would cluster together on a tree. And if they cluster together on a tree, it would be really nice for those public health jurisdictions to see that and then be able to initiate a collaboration to figure out what's going on, a joint investigation.
Okay, yeah, I appreciate that response. Thanks. I'm definitely looking forward to the open sourcing of the front end too.
Yeah.
Hi, David. Thank you for the talk. Being a small lab in DC, we're very interested in something like this. But kind of just to add on to the sensitivity issue with data sharing, you said it goes by organism and organization, but can that be, what is the de-identification if we wanted to, have hospital A, B, C, that kind of thing? Is that possible?
Yeah, absolutely. It's based on what you find useful. So the public health
user input, or how does that work?
Yeah. It's you provide it. So so if you don't feel comfortable providing hospital names that would only be visible to you and no one else in the system, then you can certainly just use hospital A, B, and C. The public health nurses at Alameda County, I think they feel pretty strongly that minimizing the number of dereferencing hops that happens is greatly helpful. So they appreciate being able to look at the tree and seeing, county, like the specific county hospital that, that they might be working with. But certainly there's no, you can use the system without providing that information.
We would love it. The hospitals we work with might not. Yeah. So, and then as far as the login, is that just like by user, or they kind of have like the hospital as a group has a login, or?
No, it's individual users. So individual users are affiliated with a group. So there's many users that are affiliated with Alameda County, and then their reports are sort of organized jointly as, anyone from that group can have full access to the reports.
Okay, thanks.
Two additional questions. The first one is, I'm curious about whether or not the Madrone hold any sort of a PHI or PII. If not, or if it is, are you guys HIPAA certified? And if it's not, are you forbid the nurse practitioners upload any PHI and PII? And the second question is, from a clinical hospital to find the patients and do the isolation and send it to the county labs and finish the sequencing, upload the data, finish the analysis, and trace back to the action, what's sort of like the rough or average turnaround time for them to act in a timely fashion?
So to answer your first question, we don't host any PHI or PII at the BioHub. So the only data that is submit are the bacterial sequences, the data of collection, and the collection sites. So those are the only data that we host. But we are working, so the EHR project that David mentioned on the second to last slide, we are working with UCSF on that project, but all of those sensitive data will be on UCSF hardware. So none of those data are being transmitted to the BioHub. So we are trying to develop like on-prem versions of Madrone, and also like the other analysis pipelines we're developing based on the EHR data. So we definitely have no plans to host PHI or PII at the BioHub. So that's not a concern that we have. And for the second question, maybe David would have a better answer for the. Oh, the second question was like how long it takes for the workflow from after they see something on Madrone and then working with the hospital.
Yeah. So once we get the sequences, the backend analysis is pretty minimal time wise. You know, we get the reports turned around in a few hours. In terms of how Alameda County's workflow goes, so they're on a weekly sequencing kind of schedule. So they sequence on Wednesdays. So, you know, and then also compliance kind of varies on how quickly samples get mailed to Alameda County Public Health Lab, right? So some facilities are super on it, and, you know, within a day of culturing, Alameda gets the samples. And if they get it on a Tuesday, they'll sequence it on Wednesday and upload it on Wednesday, and then they'll have the report on Wednesday. So it can be basically as little as 2 or 3 days. But I think more common is it usually takes them about a week or two to send the samples to Alameda. And then depending on where in the week it falls, you know, it gets sequenced on Wednesday. So yeah. So I would say, you know, total, it's probably they're seeing things on average, maybe about 2 weeks after the peripheral facility sees it.
Got it. Thank you.
Is this limited to clinical isolates, or are you looking for environmental, or it just depends on the user?
Currently, it's limited to whole genome sequencing data of bacterial isolates. One of the projects we're embarking on in 2020 is to attempt to do a version of this on metagenomic data. And so, but that's kind of an R&D project. A lot would have to go right for that to become operational.
But if we were to have like a Legionella isolate from an environmental sample that we had WGS data for, and
yeah, absolutely,
those could all be submitted.
Yes.
Yep. Okay.
Yeah. That's actually something I wanted to ask too, David and Lucy. In terms of SPID, it's just so long as we have a reference genome for it, we can do any organism our heart desires as a user?
That's right.
Okay. And that's something the user will have the option to upload in terms of a reference genome assembly, or is that something that's kind of maintained on the dev side?
Currently, it's set up that we maintain it on our end. If there was, you know, let me kind of maybe make a more general statement. You know, so as you can see on this team slide, you know, we're a small team compared to some of these other projects that are being written. So, you know, the BioHub is very passionate about trying to have an impact. So basically, you know, if we found, you know, a jurisdiction, for instance, this Washington jurisdiction, you know, that was really passionate about routinely using it and working with us, you know, we would assign resources to basically make the tool work for them. So really, you know, this is a nice opportunity to try and connect with some of these public health, some of you all, and, you know, based on the response we get and the level of interest, we can kind of titrate our resources. But certainly it would be possible to develop the capability to upload your own reference to the workflow. But currently the way it would work is you would email us and say, we want to use this reference. Yeah.
Hi, this is Logan from Colorado. I really enjoyed your talk, but I do want to kind of get into a little bit more about that, using the reference and stuff like that. So it sounds like you guys are choosing a reference per situation or per jurisdiction that's using it. Is that correct? Or are they giving you a reference and then you upload that to their...
We've been mostly choosing the references. I mean, certainly in consultation with them, but so far our users haven't had the level of expertise where they've analyzed their sequences and come to us and said, okay, we've done some sour mash sketches and we're confident that this is the reference. We've been doing that part of the workflow so far.
Okay, and as kind of a follow-up to that, then, so is there any worry about the robustness of the system in identifying kind of clusters or things like that? So like say if you had one of those cases where somebody from Alameda goes to San Francisco and then they're seeing it on their system, but then Alameda's also looking at their system, are you ever going to have a situation where one of the things will call it based on the reference that they're using versus the other one which would say, oh no, this is not? Have you looked into that?
Yeah, so currently the validation of the system is done entirely on the end user parts. So Alameda has, they have a kind of ground truth set of clusters that they've in the past called, and they check to make sure that those are still being called correctly, and they email us extremely quickly if there's if there's a one SNP difference, but we haven't seen any we haven't seen any problems with the the ability to call clusters. But I should also add that it's exactly because we haven't confronted the sort of scenarios that you're describing yet. So in other words, we've been constrained to sort of like limited geographic regions, and a relatively small number of samples where it's, it's sort of easy to pick one reference and then kind of stay there. So we will be, kind of carefully addressing these issues as we start getting samples from San Francisco and we start confronting the issue. So I guess the answer to your question is validation is currently being performed by the end users. We've been kind of getting away with that because of this relatively smaller scale. As we scale up in 2020, we're going to need to invest in kind of more systematic validation of
to add to that, we have done some testing on our side in terms of SNP calling with different references. And we obviously see some differences in terms of the number of SNPs between a pair of isolates. So we've worked with the Alameda County team to initially set very generous SNP thresholds so that we can capture a lot of potential false positives. And then over the last year, they've done their own investigations, and then they've optimized the SNP threshold themselves to a level where they have found that everything below that guaranteed to be clusters, and they haven't found anything above those thresholds that they suspect are clusters. So it's definitely been an empirical process, and I'm sure you'll appreciate that. It's not an exact science. There's so many different variables that affect what that SNP threshold should be. And so right now we've settled on an empirical approach to setting that SNP threshold.
Okay, thank you. Yeah.
Lucy and David, I actually had a question too about SPID, something that you probably mentioned in the talk, but it was kind of maybe went over my head a bit. So in terms of, you were mentioning the issue with increased volume would shrink the core genome that you're actually looking at, but something in the SPID workflow allows that phenomenon not to occur. Can you explain exactly how that would happen? My assumption here is that we have increased sample volume, we're going to have increased genetic diversity, which is why we see that decrease in core genome of which the SNPs can be called. So can you talk a bit about on how SPID avoids that?
So yeah, so SPID just aligns. So SPID is a very simple. It's arguably much simpler than a lot of the joint callers, right? So it simply aligns against the reference you know, the chosen reference, and regions that don't align just don't align, right? So a given sample will have, you know, if you can imagine the reference genome stretched out on top, you know, one of the samples will have alignments where it has alignments, and then, you know, no alignments. I don't remember if we represent those as gaps or ends. So then when two things are compared, you know, we rely on them having a substantial chunk of overlap you know, in their aligned region. So certainly it would be possible, you know, in sort of an adversarial case to have, you know, sample A that aligns only in the left region and nowhere else, and sample B that only aligns on the right region and nowhere else. And then the pipeline would fail trying to compute a SNP distance between those. But that doesn't happen because we're restricting to, you know, samples that have limited amounts of recombination, are relatively closely related to each other and to the reference.
Okay, so there's some narrowing of the sample set before it gets aligned to the reference in that way.
So sort of implicitly, just because, you know, we're choosing a reference on the basis of, you know, some health department is like, we're tracking Acinetobacter infections in our jurisdiction, and then we focus only on those samples. So just, you know, just because they're limited to a geographic region, they implicitly will, you know, will be pretty highly related to each other.
Okay, thank you.
I have a quick question. I'm just curious about the user interface, the front end, and how easy it is for the users to interact with it. So I'm just curious, like, what are the basic steps for like getting data in? I assume that's done by staff, the Alameda County lab that's doing the sequencing. So like, what are the steps that they take? Is it literally just dropping reads into a website, and then the steps for the infection control nurses to make use of the report? You know, do they have to select workflows? What sorts of steps do they have to take as well?
Yeah, so at risk of embarrassing myself, let me attempt a live demo.
Thank you. We understand if there's any sort of hiccups.
We'll edit this in post.
So by the way, I should also mention that you all can check it out. So if you navigate to HTTPS, so the S is important, //madrone.czbiohub.org, and you log in as demo, you'll be able to follow along. If we have 35 simultaneous users, that may be an all-time peak. So we'll also be stress testing it. So just to kind of answer your question, to go through the workflow. So the first kind of step is the upload interface, which looks like this. So can you see my screen? Hopefully you can.
Yep. Yeah, we can.
So it's a two-step process. So you first prepare a sample information spreadsheet, which is a simple Excel spreadsheet that just has one line for each sample. And you basically, like Lucy mentioned, you provide the sample collection date, the sample collection location, as well as you specify the FASTQ file that you're going to upload for that sample. So, you know, maybe that sample information spreadsheet has one thing in it, maybe it has 100 things in it, and you just, you know, you prepare it 1 row per sample. So then you take that Excel spreadsheet and you just drag and drop it here on the top box. So then in JavaScript, we parse that spreadsheet and we pull out only the columns that we expect. So in particular, this is an attempt to prevent any accidental upload of PHI. So we, you know, if you have your columns with that information, but for some reason you also have patient names on the bottom right of the spreadsheet, those won't be pulled out. It'll only pull out those known columns. It'll also, I guess maybe I could actually try to really do this. Let's see.
Send you an example spreadsheet.
Oh, you did? Okay, I think I might have one also.
Hey, David, one quick question.
Yeah.
Do the submitters have to have certain QC requirements prior to submitting, you know, coverage, that type of stuff?
Currently, QC is part of our backend pipeline. So we take a look at it and, you know, we make sure it doesn't look insane. And if it looks insane, we reach out to them, but it's not, we do it on our end. We don't require
What constitutes insane? Just you know, out of curiosity.
So what constitutes insane would be like coverage less than 20x, I would say. So we seem, you know, because if we call Snips with 10x with 10 reads, then having about 20 is about the lowest we're willing to go. You know, and we also look at, you know, at the sequencing Q values just to make sure that, you know, something didn't go wrong with the Illumina run. This is short read data. I don't know if I mentioned that before, but our pipeline's set up for short read data right now.
Thank you.
So coming back to the upload interface, so what's visualized here is after I've dragged and dropped. So I've dragged and dropped an Excel spreadsheet. It's been parsed, and 5 samples were in this spreadsheet, test 1 through test 5. So it pulled out a patient de-identifier. We use this de-identifier to detect self-clusters, right? So if you submit multiple samples from the same patient, you can give them the same patient de-identifier, and then we don't call an outbreak cluster if you just have repeat samples of one patient that have 0 SNPs or 1 SNP, right? The FASTQ file name is the FASTQs that are going to be uploaded. The facility name is, you know, whatever facility name you're comfortable providing, the specimen collection date, and the source. So the idea is this is parsed and visualized for you, and you take another look at it just to make sure it looks correct, and then you click this blue button to submit the information to us, submit the sample information to us. And once you do that, down here, it'll ask you to drag and drop the FASTQ files. So then you drag and drop the FASTQ files, and they get sent to us for analysis. So that's what the upload interface looks like. So once the samples are analyzed and visualized, they're added to a report. So here the report is called Demo California Klebs Pneumo. So you could have multiple reports in your jurisdiction, maybe one for Klebs Pneumo, one for Acinetobacter, one for something else. So you click that to select the actual report, and then you see that you see the visualization from the video. So you see how many samples were recently added to the report, as well as, you know, so you can visualize which samples were uploaded recently by clicking that, and you can see the suspected outbreak clusters that were called based on the SNP threshold. Down here on the bottom is a threshold slider where you can adjust the SNP threshold that is in effect when you interact. As you interact, you can visualize the SNP distances between various samples. So you see for this cluster, the SNP threshold was 0 SNPs, so that was why it was called a cluster. But then if I look at some of these other ones, this one is 30,000 SNPs away from the one that's selected below. And this one is also 30,000 SNPs away.
And David, so on the upload on that data sheet, is that also where people would put like the coordinates for that mapping feature there on the bottom right?
Yeah, they would put the facility name in, and then the database has lat longs for that facility name, so then they could be visualized on the map that way.
So when we set up a new user, we would get a list of the facilities in their jurisdiction and their geographic locations. But that's not they don't need to submit the location information. So if they're not comfortable with submitting the coordinates or like the facility name, we can still accommodate that. So the pipeline still works exactly the same. It's just like the facility names will be censored and they wouldn't appear on the map.
Yeah. Okay.
Hey, Lucy and David. This is really great. I really like the interface and, you know, focusing locally. But I will be curious to see, as you expand this and bring it to more public health labs, how the reference-based and SNP-based approach scales. Because the other 2 big efforts that I know about using SNP-based approaches, you know, with the PulseNet and, you know, with NCBI, both of them had to transition away from a SNP-based approach to a whole genome MLST approach because of scaling issues. And, you know, you'd make whole genome MLST clusters, and then within the cluster, they'll look at SNP analysis within that cluster. So I'm curious, have you given a lot of thought to, you know, if this becomes more and more adopted and scales and bigger and bigger, how your approach will scale?
So you know, I guess I would say that you know, we as an organization, we feel that the computational requirements are pretty modest for this backend. So we have other projects that have much higher computational requirements, right? So definitely, for the short to midterm, like we're fine on doing the compute, and we view that as kind of a valuable benefit, like to whichever health departments want to partner with us in this early phase, we're basically able to give away compute for free. Certainly, we probably can't be doing this analysis for millions of samples a year. There's obviously some upper limit, but no. So to answer your question, we haven't thought of the ultra long term scaling yet. That we would envision that as kind of a joint conversation. Like if this really became a tool that like public health was keen on adopting in the United States, we would want to probably discuss forming some kind of nonprofit to take this over, and then maybe the public health departments would, contribute some fee for the computation that would be, just to sustain it, something like that, right? But that would be a very long term, sort of like moonshot success. Definitely for the short to midterm, we can do the compute for 10, 20, maybe even more health departments participating in this.
I mean, we are considering solutions to the problem of scaling and the trees getting bigger, like
for visualization.
Yeah, for visualization. And I think at some point we'll explore like more k-metre-based approaches as well for initial clustering. So we've had some success with the split k-metre approach for some of our other projects. So that's also like avenue we'll be exploring in the coming year.
All right, this is Sean from Minnesota again. I guess
kind of at the top of the hour, so I just wanted to give
one quick question.
All right, go ahead, Sean.
So this is maybe beyond Madrone, but lots of the CRE transmission or antibiotic resistant might based on the plasmid. Do you guys have any thoughts about the potential plasmid transmission between different species?
Yeah, that's a great question. So we're currently developing a algorithm to flag potential horizontal gene transfer that uses information. So ideally, what you would do is you would get circularized plasmids, and then you would compare sample A against sample B. Is the circularized plasmid the same plasmid, right? Or is it highly similar? But of course, with short read data, you can't really do that, right? You don't have the capability to sort of resolve those repetitive regions and circularize the plasmids. So we have an algorithm that's in development right now that we're going to be writing up and publishing soon that flags horizontal gene transfer on the basis of a tangled assembly graph. So basically, starting with an assembly graph and looking at the assembly graph context near an antibiotic resistance gene, we feel, on the basis of what we've seen so far, that we can reasonably flag potential horizontal gene transfer. So that's a preprint that we'll be finishing probably in the first quarter of 2020, and then we hope to operationalize and implement that algorithm in Madrone subsequent to that next year.
Wow, that's awesome. Thanks for that.
So the way that would look is it would basically scan pairs of isolates, and if there was suspected horizontal gene transfer, it would flag it for the user in a separate user interface.
And David and Lucy, I don't know if you guys have time after I close this call to stick around. I think there's even a couple questions coming in the chat.
Sure, yeah.
Okay, so and one last thing too, for this pilot project, can you talk about how participants can be a part of this and register, and then we'll close out the call.
Right. So the one thing Lucy and I would like to advertise is that the BioHub is really committed to trying and replicate the success. So we the things we can do is we can provide the compute, and now we can actually provide a sequencer. So, we're aware of the fact that at the state level, many labs have sequencing capacity. But if there is a jurisdiction, maybe either a state jurisdiction that doesn't have a sequencer available for genomic epi work, or a local health department that has a lab that would like to demo a sequencer, and epidemiologists that would like to use this tool, we're starting a pilot program where we would essentially loan an Illumina sequencer out in order to participate in this program. So if any of you that are listening have acute communicable disease controllers, epidemiologists who would like this tool, and a lab that would be willing to do the sequencing, but you just don't have a sequencer, please get in touch with us, and we can talk about the possibility of doing this pilot sequencing project.
All right. Thank you, David and Lucy. I'm going to go ahead and end the recording for those of you guys who need to make the next call. But thank you for that. So I'll keep the line open for another 10 or 15 minutes so we can get the last few questions in. Thank you, everybody.
Thank you. And just in terms of getting in touch, you can email all of us, Madrone@czbiohub.org.
Thank you, guys.